Hello, everybody, and thank you for attending today's seminar. I'm Kevin Beck. I am a senior manager in the commercial operations group of Milteney Biotech, working in our Lentigen technology division. I have been with Milteney for nearly 7 years, over 7 years, in a variety of commercial roles, and I've been with Lentigen for coming up on 2 years. Today, I will be talking about accelerating clinical development and commercialization with a lentiviral vector platform manufacturing process for cell therapy. I'll start my talk by introducing you to Milteney Biotech in general, and then specifically our Lentigen division, a brief introduction to lentiviral vectors before proceeding into our service offerings. I'm a firm believer that when you choose a CDMO to partner with, that you shouldn't just find somebody that can make a good product for you, but can also help you get to the market and into the clinic. And so I will also be reviewing our regulatory support and our plans for supporting commercialization. So hopefully many of you have heard of Milteney Biotech before. For over 30 years, we have been a leader in the global design, development, manufacture, and integration of products to empower the advancement of biomedical research and enable discovery in cell and gene therapy. Some brief facts on the company. So as I said on the last slide, we've been around for a little over 30 years, established in 1989. Our portfolio has grown quite broad. We're approaching 20,000 products total. But I'd like to call attention really to some of our clinical aspects. So every year, nearly 2,500 patients are treated with cell products that are derived from our technologies. And in total, over the course of our history, that has been a sum total of over 55,000 cell therapy procedures. And so this is a field that we've been invested in quite a long time and have quite a lot of knowledge and expertise. Specifically, I sit in our Lentigen technology division located in Gaithersburg, Maryland. This is our original Gaithersburg site. I'll talk about a second site a little bit later in the talk. This facility is approaching 40,000 square feet with 2 ISO 7 clean rooms for lentiviral vector production that meet all US and EU clinical requirements. And in total, we have a capacity of up to 80 lots a year out of this facility. As our name might imply, in our Lentigen division, we exclusively use lentiviral vector technologies. We like lentis over other vector systems for a few reasons. One is their efficient and stable transduction of both dividing and non-dividing cells. We like their high gene payload capacity, allowing for some very interesting constructs like duocars and even tricars. Downstream of our production, when you move into your cell product, the mechanism of integration of lentiviral vectors generally yields a very high transgene expression. And finally, we like that lentis can integrate very nicely into the Clinimax Prodigy T-cell engineering workflow. So we're able to provide our vector in vials or in bags that are purpose designed for sterile welding directly onto the Prodigy. So now I'd like to talk a bit about our service offerings for lentiviral vector production. So Lentigen has its own long history in the cell and gene therapy field. So we were founded in 2004 by Boro Dropulic, who remains our chief scientific officer. Some of the earliest work by Boro and team went on to support the first in-man clinical trial that used lentiviral vectors, showing their safety in the clinic. Lentigen has then gone on to develop a number of our own assets, and you can see some of the usage of those denoted on the timeline. In 2014 was an inflection point for Lentigen's life cycle by the acquisition of Milteney Biotech with a real synergy in the interest in cell and gene therapy. That was borne out a few years later by integration of the T-cell engineering process on the Clinimax Prodigy, and you can see that those are getting used in the clinic. In terms of our own in-house developments, in 2017, we brought our 50-liter serum-free suspension process online in GMP. I'll tell you a little bit more about that later. A few years later, we scaled up. So in 2019, last year, we brought our 200-liter GMP serum-free suspension online. And we've also begun our move into our new facility that I'll talk about a little bit later in the talk. And the key purpose of that new facility is to support commercial manufacture of lentiviral vector, and we expect to be ready for that in 2021. We focus exclusively on third-generation lentiviral vectors. So all lentiviral vectors are derived from an HIV-1 genome. Over the course of the generations of the the lentiviral vector production systems, the genome has been stripped down to its core components and separated onto multiple plasmids. Ultimately, this reduces safety concerns, so you reduce your risk of generating a replication-competent lentivirus. In our history at Lentigen, we have never generated a replication-competent lentivirus from our third-generation system, and to my knowledge, there's not a positive case in the field. In total, the way that this works is that the genes are separated onto 4 plasmids, 2 we would call our packaging plasmids, which contain some of the basic machinery needed for making the virus. There's the transfer plasmid that contains the client's gene of interest. This can be an R backbone, which is optimized towards our system, or a client could provide their own backbone. And then finally, there is the envelope plasmid, which confers the pseudotype. We use a VSVG, which confers a broad infectivity into many cell types. As a forward-looking approach, other envelopes may be available as well. As I said, we use a serum-free suspension process. So our legacy process was an adherent process, and all legacy processes are adherent. We think the serum-free process has some real improvement. So this is a chemically defined process, so it improves the CMC profile. It increases the scalability of the process overall. So at our maximum capacity in the adherent, we were working at a 32-liter scale. And as I mentioned, we can make a 200-liter batch with serum-free suspension. And taken together, the serum-free suspension in our hands generally increases both volumetric and total yields while providing a similar functionality of the cell product. So I'll show you some data to address that last point here. This is data that was produced using a model car that we use in many of our process development studies. You can see there's an adherent batch, 2 serum-free suspension batches at 50 liters, and 1 at 200. Things that I'd like to call your attention to is that the harvest titers tend to be about double in our hands in our serum-free process versus our adherent process. So we tend to get a harvest titer on the order of 10 to the 7th. And then we go through a 100-fold concentration process, yielding final titers generally on the order of 10 to the 9th. Taken together, of the higher production and the higher scale, you get a substantially higher absolute total yield. This is based on titration units as determined by a qPCR assay. So ultimately, the serum-free suspension process reduces the cost per dose per our clients. When we made the change to serum-free suspension, it was critical for us to show comparability of the products. You can see here that we've taken the same car. In this case, it's a model CD19 car. We have transduced cells using the same MOI, and we get very similar CAR expression. Critically, the graph at the top right is from a cytotoxicity assay, and there's a blue and a green line directly on top of each other. One represents the adherent CAR, one the serum-free suspension CAR. And so you can see the cell functionality is very similar. In terms of the way our platform process works, we separate into sort of standard upstream and downstream functions. In our upstream operation, we keep a rolling culture going. There's, of course, limits on number of passages and that sort of thing, but this allows us to minimize the time that it takes to seed our production vessels, because we can seed with some larger volumes. As the production vessel cell density gets up to the appropriate amount, we transfect with the 4 plasmids that were described previously, and then we harvest. From there, we move into our downstream process, which involves clarification and concentration, purification by chromatography, and then we sterile filter and formulate on our way to final fill. So that's how we do things. In terms of what we are able to do for our client, I think this slide really accurately describes what a normal project might look like for one of our clients. So we generally start in the sort of development and feasibility phases, not discovery so much. We're really not set up very well to support that. But we can make small-scale research-grade batches. These are non-purified vectors, but allow our clients to put multiple constructs in rank order, comparing different sequences, regulatory elements, promoters, et cetera. We can also learn at this stage if there are any major challenges associated with the client sequence that might need to be addressed from a molecular biology perspective before moving forward. After a client has picked a construct from the research-grade vector to move forward, we generally recommend we make our next stop at our demonstration batch, which is our 4-liter scale. And this is our smallest scale that has a full downstream process in place. The data that we get out of the 4-liter batches, and this generally yields somewhere around 30 to 35 milliliters of material for client use after all testing. The data that we get out of this is very predictive of the titers that we will get from the 50-liter or 200-liter GMP scale. And I'll show you some data to address that in the next couple of slides. So from there, depending on the client's needs for experimental studies, they may choose to have some larger scale preclinical batches. Most of our clients skip engineering batches. We have a well set up process, and so we generally don't need to do an engineering batch to know that we'll be successful in GMP. And so that's one of the advantages of moving with our platform, is that you can go from a 4-liter right into GMP and really have a strong expectation for what your production will be from GMP. In terms of some data to address this, there's another model car study. We're comparing a demonstration batch. This is the 4-liter batch in orange versus both 50 and 200-liter serum-free suspension batches. You'll see two different types of titers represented here. There's a functional titer, which is a qPCR assay, and a physical titer, which is a P24 ELISA, showing you information on the number of particles that are around. The N's are very small on this study, but I hope this begins to show you that the data that we get at 4 liters is quite representative of what we would get at the GMP scale. Expanding this a little bit further, here's some client data that's been de-identified. A few of these products are made multiple times, so you'll see a letter appear multiple times on the axis. I think there's a few interesting things that you can see from the qPCR data of transducing titer units per mil here. First off, we tend to make a harvest titer on the order of 10 to the 7th. We tend to get that up to about 10 to the 9th after our concentration process. We're quite consistent within a given product, so if you look at the B's versus comparing those to the D's or the E's, you can see that each product has its own profile, but we tend to be consistent within product. But critically, there is a sequence dependent. So it is a product-specific result that we might see. As I said, when you choose a CDMO, you shouldn't find just somebody that can make the best product for you, but somebody that you're comfortable with can get you into the clinic and ultimately to the market. I won't review this slide in detail, but as I started in my introduction to the company, Miltenyi has been in the cell and gene therapy business for 30 years. And so we have quite a bit of experience supporting INDs and IDEs with the FDA and many clinical studies in the EU and the rest of the world. Accordingly, we also have a fairly mature quality system that helps with those sorts of things. Specifically, in terms of our regulatory support for lentiviral vector services, we do use the same global regulatory support and US regulatory support teams as the rest of the company, so we leverage their experience. We have biologics master files in place with the FDA and with Health Canada. When a client product is made, we're able to amend that master file with any product-specific detail and reference all of the process-specific detail that's kept confidential, and then provide a client with a letter of authorization, allowing them to link our master file to their application. In regions without master files currently in place, such as Europe and Asia, we generally provide a product information file, which provides the client with high-level non-confidential information, allowing them to fill out their dossier, and then we could provide a direct submission of any confidential information to the authority. Our biggest push recently at Lentigen is moving towards supporting commercial phase vector. And this has taken a lot of effort on our part. So I think the three things that we've really needed to get ready are our facility, some of our quality procedures, and then finally, we need to get a few products ready. So the client's product needs to really be prepared to get into a commercial phase. In terms of getting our facility ready, our facilities management working quite hard to bring our second Gaithersburg site fully online. It is partially open. I'm sitting in it now. During its full opening phase of phase 1 of its manufacturing suites, we'll bring 4 more ISO 7 suites online and have all of the same sorts of capabilities that we have at our original Gaithersburg site, and also duplicated in Tetterow, Germany. In terms of our new commercial facility, it's located directly across the street from the original Lentigen building that I showed previously. The total area is approaching 145,000 square feet with all of the operations that you might expect in terms of R&D, process development, manufacturing, and back office support. I mentioned we'll have 4 suites that open up during phase 1 of the manufacturing area, and that will add an additional 160 GMP lots per year to our capacity, in addition to the 80 lots in the original facility across the street. So that's 240 lots per year that we could produce in Gaithersburg. Further, we do have an adjacent lot that's also quite large. It's approximately 5 acres with room for another 100,000 square foot building to be used as needed. Having been a leader in cell and gene therapy for a long time, we have quite a mature quality system. We are ISO 9001 and 13485 certified. You can see some of the Code of Federal Regulations that we follow here and some of the certifications that we've received. Despite this, we still need to mature even further in order to be prepared for our commercial phase. And in part, that's because we apply a 6 systems approach to quality management, where quality really sits at the center of all of our operations and are involved in nearly everything we do. This is not an extensive list, but in terms of some of the things our quality team has been doing to prepare for commercialization, as we've migrated from a paper-based to an electronic document management system using master control. We've implemented a CAPAs and issues management module, change management systems. We've been auditing many more of our suppliers for quality and putting quality agreements in place with them. We're moving our analytical methods from qualified to validated. Our GMP trainers working on training our cross-functional teams on pre-approval inspection readiness. And as part of that, we've also performed a formal gap assessment against the US and EU commercial regulations and are preparing for some third-party mock inspections before the authorities have their chance to audit us. So those are our preparations, and I'd say the final preparation is something that really needs to involve our clients. In terms of a product development lifecycle for lentiviral vector, we apply a quality by design approach. You can see that illustrated, the lifecycle illustrated in the image on the slide. So really where we sit right now is sort of between these process development and process characterization boxes. As a client has their product, so we need to characterize our process in the context of the client's production. So we would go through a series of process characterization studies, ultimately yielding our final control strategy. In terms of how we approach this, as I said, we do take a QBD approach. We'd work with the clients to define the critical quality attributes. We'd start with a high-level risk assessment, establishing a linkage between the CQAs and any unit operations. Follow that with a deeper dive, establishing a linkage between process parameters in the CQAs at a lower level risk assessment. From there, we were able to identify any process hotspots in need of characterization, and then we'll take a design of experiment approach to design experimental studies that will ultimately yield the information that we need to define the process control strategy. We think this process takes somewhere around a year to complete, and at the end of that, we'd be ready for process performance qualification runs, and then ultimately the client should be ready for licensure. Just to wrap up, I hope I've shown you some information that leads you to think that you can use a platform process to accelerate from development into manufacture. Ultimately, this will minimize the time to the clinic, the time to first in human, or the time to market. It can be a very cost-effective strategy for your organization in that developing processes that fulfill regulatory requirements is both time and resource consuming, and we've already invented that wheel here, so it's not something that you need to do necessarily. It also helps reduce the complexity of required tasks like process and analytical development. Again, we can take advantage of economies of scale and leverage our experience as a specialized CDMO to help your organization. So I thank you all for your attention, and I'm glad to take any questions.